all in all, much better than the previous times. Less work for me, less leg. And I mentioned the label, the very first heading, your name, the course, and the date. It should be single space. Okay? I didn't say that. I did write it down on everybody. Every other thing, it should be double space. And I mentioned different font, and I don't know the difference between style, font, and size. It should be the same font, right? And the same style, size. Right? Every single summary and or formal lab report should be exactly the same. Font, the same font, and the same size. But for the headings and the subheadings, okay, they have to be a different style. Save the italicized form for the scientific names. The scientific names of any organism is the genus and the species. And if you want to take biology too, you'll learn that the genera name and the species name, it should be a descriptive in Latin or Greek, okay? Describing that. For instance, Megaptera novinglia, the humpback whale. Okay? Mega, it refers to large. Ptera, P-T-E-R-A, ptera, it means a wing or feather. So Megaptera literally refers to long fins or wings, and that's the genus name. Novinglia, that refers to New England. Okay? So the long-winged New Englander, okay? The humpback whale, okay? That we see in the North Atlantic Ocean. So again, the scientific names of any organism in biology refers to the genus name and the species. My point here is that save the italicized style for the scientific name. You can use underline, bold and underline. Okay. I think I started a couple of them. Don't do state what you've done. Okay? Don't direct in your summaries and or your formal lab report. For instance, place test tube in a hot water bath.
So is that more like like a tense issue? Like place would be instructing versus place.
Oh, no. It was placed. Okay? I have, don't use a lot of I and we. you can use one or two, but every other sentence should not say, I placed 5 milliliters of distilled water in a test tube. And definitely don't put direct, like you're writing another lab right, thank you. Direction. And all in all, they were very, very highly organized. That's my game plan. I don't really care too much about the content at this point, other than the technical aspect. Okay, do we have all the figures? Okay, number one, again, all right, 1 and 4, this thing. Number 1 should be D, nucleus, which is, not true. They don't have a nucleus, so D. Which of the, B, plasma membrane, nucleus, and mitochondria are shared by all eukaryotic cells. All the other plasma membrane, cell wall, no, only plant cell walls. Plasma centrioles, no. Chloroplast, no. B, the only. The interaction of 2 polypeptide chains.
Quaternary.
Right, quaternary structure. 2 or more. I did that in the hemoglobin molecule. So 2 polypeptide chains interacting. That should be, number 3 should be D. D, right, C this time. Again, I won't, if you got it wrong once, I'm not going to mark you wrong twice for my mistake. Number 5, B, uracil. Uracil is not found in DNA. Thymine, well, it is. Uracil is in RNA. Conjugated proteins out of these choices. C, chromoproteins. Okay. Color. 2 or of the 4 structures of protein. Primary structure, delineation or straight line of polypeptide chain. Secondary structure, pleated sheet or helical molecule. Tertiary structure, incorporated in globular proteins. Stabilized by hydrogen bonds, and or you could have said quaternary structure. Again, 2 or more interactions of the polypeptide chain. So the 2 of the 3 generalizations of the cell theory. Okay? The biogenic law states that all cells come from pre-existing cells. You could have said the cell is the basic unit of life, as we know it. And what else?
Well, cells are
One or more, every organism is composed of at least one or more cells. It makes sense for the basic unit of life. And this should have been a gimme. Terpenes. What are terpenes?
Fat soluble,
Right, fat soluble
Vitamins.
vitamins.
exciting.
You need the, oh, well.
Me too.
Okay, I left, oh. This, maybe one or two questions on the folding of for the exam. Denaturation, even though I say high heat and pH, it could be low pH or high pH. Acids will denature proteins as well as bases. Right? Definitely heat, high heat. And we're gonna do the enzyme lab. Again, you should be aware that all enzymes are proteins. Nucleic acids, the RNA and DNA, nucleotides, one inorganic phosphate group, one pentose or 5-sided sugars, and one of these 5 nitrogenous bases. In your, you see your cell, you're thinking RNA, and if you see thymine, you're thinking DNA. Adenines and thymines will come together, join together, and cytosine and guanine will come together. Uracil and adenine in on animal cell and the plant cell. I left off. Passive, no energy is required. It goes through with the gradient. Right? If you have, and that's the idea of a cell, the beauty of a cell. You're having membranes, so you can divide high gradients to low gradients on one side. So on the high gradient side, it wants to be equal, right? And it can't easily go through that membrane. And that's why when those gates are open, those molecules or atoms are constantly want to come in. So those gates open and they flood in to equalize the concentration gradients on both sides. Active transport, it goes against the gradient. So you need some type of energy. simulation, and again, I'll talk more about, today, bulk transport, okay? Another type of transport. But before I forget, good news, tomorrow, I don't want any lab summary, okay, for tomorrow's lab. Okay? But you will be responsible for a formal lab report for tomorrow's lab. The fifth lab, osmosis and diffusion. Okay? So if you can work on, give you plenty of time. If you hand it in to me on or before this due date, the 2nd of July, I'll look it over and do the same thing I do with your lab summaries. I edit them, and I'll give you a grade. If you want to keep that grade, that's fine. But most students are not happy with the grade that I give, so they can do a rewrite. Okay? So if you hand it in before or on this date, I'm going to give you another date. Okay? If you don't hand it in for a rewrite, okay, that's it. Okay? This will be the actual due date. Okay? Again, if you don't hand it in for a rewrite, okay, the 19th of July will be the final, all set with that. And that, by handing it in for a rewrite, that's one of the prerequisites to be exempt from the final exam. Okay, questions? All right.
What makes a lab report different than a lab summary?
More in depth.
Or-
Okay. Review the lab. Oh, I have this. I'm talking about all this stuff and okay.
So how long does the lab report need to be then, like 5 pages?
As long as you get your information across. John, you had a question?
I was just asking, for the lab reports, how long do they need to be, like 3 to 5 pages?
Yeah, with your, this is, I'm surprised this is, labs. This is, in your, the back of your, oh, right. I didn't print all of this, but it is in your, online. Okay? I did not print, I saved a few dollars. Online, okay, I posted all of these online. And this is the differentiation, okay, between a lab summary and a lab report, okay, a formal lab report.
Literature cited, so we have to. Research
Right,
outside
at least, one or two books, okay, and and one or two online things. So anyways, I give you a little bit of how to write, introduction, okay? Introduction, the main difference is, between a lab summary and a lab report, is the, materials and methods. I did not want to, mention, in a summary, but in a formal lab report, yes. The results, all of your data, tables, images, go in the results section. I don't want to see any images, tables, or referring to any images or figures or tables in the discussion or the materials and methods or the conclusion, just in the results section. The materials and methods do not just list the materials that, like a shopping list. Okay? In italicized form, you can put all of these materials, not in a list, but in a logical sentence. To demonstrate diffusion of a liquid in a liquid, 1 milliliter of malachite green was added to a 250 milliliter graduated cylinder. So I got all of these, most of these materials in one sentence. And that's the beauty of the scientific writing. The last sentence of your introduction, and I didn't mention this, and I don't mention this for the summaries, but for the formal lab reports, the last sentence or the last paragraph in your introduction should include the your hypothesis. Granted, these experiments that we're going to be doing tomorrow, have been proved over and over again. So, in lieu of your hypotheses, just write the purpose of this, or the intent of this paper is to demonstrate the diffusion process, biological process of diffusion, and plasmolysis and turgor pressure. Okay? That I'm gonna see, and you're gonna see tomorrow, in lab. Okay? So again, if you want, I can make hard copies of these and give you that. Actually, tomorrow I think I will make copies of these. And then in the just the results section, your tables. Again, I mentioned this also in your summaries. They have to be numbered, okay, the figures or tables or images, okay? And then descriptive label, okay? And you guys are familiar with that in your summary. But again, just state the facts, the results that you got. Don't interpretate the results in your results section. That's why you need the discussion section. So another section should be a discussion section. And this should be the meat of your paper. Most of your paper. So the discussion should be at least 2 to 4 pages. 2 to 4 pages for the discussion alone. 1 page for, at least 1 page just for the introduction. Just introduce the key aspects. And only the materials and methods and the results section could be subsection with every single experiment. We're gonna be looking at dialysis tubing, cutting, making a mock cell, and fill that mock cell with concentrated starch solution, and then secure both sides of the bag of dialysis tubing. And then bathe it in a large beaker of water filled with potassium iodine solution. Okay? So we know now that ever since the chemical lab that the potassium iodine is a good indicator for starches. Okay? So if you see the solution on the outside turning black, okay, you know that the starch grains were able to penetrate through that dialysis tubing. Okay? But on the opposite side, if you see the bag or the mock cell turn black and the potassium iodine solution was still that yellowish color, you know that the opposite was happening. Diffusion process. So again, plasmolysis, okay, could be a subsection. Dialysis tubing, that could be a subsection. Okay. But again, only in the materials and methods and the results, you can do subsections. I don't want individual introduction, materials and methods, results, discussion for every single activity. Okay? Just incorporate every single, I think we're going to do 4 or 5 different activities tomorrow. It's in your lab somewhere.
So the discussion section, what subsections?
I'm sorry?
The discussion?
The discussion, no. It should flow nicely.
It's
Your
like one big
discussion.
paragraph.
There's been a lot of run-on paragraphs. I don't like run-on paragraphs. Every single topic, okay, should be indented or in another paragraph. But, I don't wanna see subsections in the introduction, and or the discussion. And surely not in the, conclusion. Just concluding the experiment. Okay? Drive home all the key points of, plasmolysis and turgor pressure and everything that are the diffusion process. Short and sweet. Do not introduce new topics in your conclusion. I hate it when students do that. And so that if it was that important, it should have been another incorporated in the discussion itself. Question back there? Okay, we all set for, I'm glad
You said this is a blackboard?
yeah, and tomorrow I am gonna give you a hard copy.
Okay.
I'll make copies of this here. Oh, don't use this. This is, I made up this data. So, I've seen this in some of my, so and I know by now that it's not gonna work. That's right, yeah. So don't, yeah. I just use this as an example. In my head, I just, right, yeah. Oh, something similar, right. You're gonna see a gradient and then a sharp, but I'll let you figure that out. Okay, we all set with that? Yes, no, maybe?
Thanks.
Okay. Passive energy goes with the gradient. Active transport goes against the gradient. And again, I'll be talking more about bulk transport. Bulk transport, I showed you that video last time with the vesicle, with the motor protein. Yeah. This literally bilipid layer. This is considered an organelle within the cell itself. Semi-permeable, selectively permeable. What does that mean?
Some things can go through.
Some things can go through where